Hi everyone, this is Nima with the White Coat Club. Welcome back to our YouTube channel. Today we're going to be talking about skeletal muscle and its contraction. But before we do that, I just had a couple updates that I wanted to let you guys know. As you guys know, I was part of the White Coat Club at University of Washington, but I have recently graduated. But I will be keep making these videos for you guys just because it's a hobby of mine and I truly love educating everyone. Today we're going to be talking about skeletal muscle. Obviously, as the title suggests, I'm going to give you a little short intro on what skeletal muscle is, and then we're going to talk about its contraction. And I have a histology slide that I could show you at the end, just so you can see what it actually looks like under a microscope. So first, let's talk about what skeletal muscle is. If you think about any muscle that you use on a daily basis, like your bicep, your tricep, your quads, your gastrocnemius, your hamstrings, all of those muscles that you actively use every day and you contract without even knowing, those muscles are your skeletal muscles. And skeletal muscles have specific characteristics. So let's talk about those real quick. The first characteristic that kind of distinguishes skeletal muscle from cardiac muscle and smooth muscle is it's multinucleate. So it has many nuclei within the cell. So we're going to talk about that real quick. So it is multinucleate. It is also a striated muscle, and we'll see why that is. It's due to its sarcomeres. But let's write that down as well. Striated. Now, if you think about your muscles, you actively can move them, but it's a voluntary movement. It is not like your muscles in your small intestine that move by themselves and make that gurgle noise that you hear every day. It is something that you contract actively. So if you were to move your arm up and down right now, you're voluntarily moving it. So it's a voluntary muscle. And to do that, you actually need to use your nervous system. So it works only by nervous system activation. So we're going to do nervous system activation. Now, why I say nervous system only is because obviously your heart can beat without a nervous input. It has its own pacemaker cells that generate its contraction, and we'll talk about that maybe in another video. Same thing with your stomach, like during like a heart exercise, your nervous system doesn't really like tell it to actively like move it. You don't voluntarily use it. It actually like shuts down during working out or something so that blood can actually go to your other parts of your body, like your muscles. Blood flows everywhere else except for your like digestive system. So those are some ideas for later videos, but for now we're just going to be talking about skeletal muscle, and these are its four I guess characteristics. And one more thing that I want to add is that it is an all or none contraction. So we're going to write that down. I apologize for my bad handwriting. So what I mean by that is when you release these neurotransmitters, you can't just like partially release some and halfway contract your muscles. When you move your voluntary muscles, you actually have to contract all or none. You either move or you don't. You don't half contract or anything like that. Those are some characteristics of a smooth muscle, which has graded contraction, which, like I said, we'll talk about in another video. But these are its 5 characteristics that I want you to know about. It is a multinucleate, it is striated muscle, voluntary, it only works by nervous system activation, and it has an all or none contraction. So let's talk about how it actually contracts. So as you guys might know, anything that happens in your body requires a nervous system input or an action potential. So I'm gonna make like a lightning bolt, and as we can see, this is a synaptic cleft. We have our presynaptic neuron, and we have our muscle cell. So let me actually go to a laser pointer if I can find it. I actually can't find it. Oh, there we go. So yeah, this is our presynaptic neuron. This is our muscle cell, and an action potential travels down as the arrow suggests. And we have these vesicles that contain acetylcholine, which is a major neurotransmitter when it comes to skeletal muscle contraction. And all these vesicles contain acetylcholine, and we have our synaptic cleft right here. And as you guys can see, as this action potential goes down, we have a depolarization which causes these vesicles to merge with the membrane and release these neurotransmitters. Then on the muscle cell, we have these ligand-gated channels, which usually allow for sodium to go in. So these blue circles that you see are most likely sodium. It could also be calcium, but we're just gonna stick to sodium for now. And once these guys are in the muscle cell, which is known as the sarcolemma, they cause a depolarization and a secondary action potential that actually travels through these tubes further down into the sarcolemma, and these tubes are known as T-tubules, and they all respond to voltage change as well. There are certain channels over here that respond to that voltage change. I'm just gonna make it over here because it's not over here, and these guys are known as ryanodine channels. And what they do is they are adjacent to a sarcoplasmic reticulum. And what sarcoplasmic reticulum does is it stores a lot of calcium in the cell. Once there's a change in voltage, these guys are gonna open, causing calcium, which has a high concentration over here in the sarcoplasmic reticulum, to release into the cell. Now, once this happens, calcium needs to bind to something for the muscle to actually contract. And what they bind to are these guys right over here. And these guys are known as troponin. And this little red guy is our calcium. Once troponin is, once calcium is bound to troponin, it causes a second thing to happen. And if you guys look at our diagram over here, we have our thin filaments, which are our actin molecules. We have our thick filaments, which are our myosin molecules. There's a blue line right over here, which is actually known as tropomyosin. And the function of the tropomyosin is basically to prevent this myosin head to attach. So what does troponin do? When calcium is bound to troponin, it causes a conformation change, which actually pulls this tropomyosin away so that this myosin head can actually bind to these yellow actin molecules. And we're actually seeing that right over here, where this troponin is going away and this actin molecule is bound to the this myosin head, my apologies, bound to the actin molecule. So we know now that calcium is needed for muscle contraction. But is that all it needs for the myosin head to just bind to the actin molecule? If you said no, you're probably right. And why that is because just like anything else in your body, you require ATP. So what role does ATP play over here? So ATP is needed for this myosin head to actually bind to actin. So what happens is, in its unbound position, ATP is bound to the myosin head. This causes the myosin head to just be charged, not actually connecting to that actin head or actin molecule. Then what happens is this ATP gets hydrolyzed into ADP, gonna make it a little better, plus an organic phosphate. And this is what actually causes the ATP, the myosin head, to bind right over here, and actually causes the muscle to shift or contract. It brings in the actin molecule closer to this M line, which I'll show you guys right over here. So this is a sarcomere. It's a contractile unit of skeletal muscle. We have our thin filaments, or actin filament, right over here, and then we have our thick filaments right over here. So right now this is in a relaxed position. As you can see, there's no bounding over here. But if there was any binding, the actin filaments would get pushed towards this M line, which, if you want to make it easier, I kind of say that it's the middle line because it's right down the middle. So that's what causes the muscle to shorten or contract. And that's basically it. The cycle needs to repeat itself. As long as there's calcium available, it'll always bind to troponin, move the tropomyosin, cause the myosin head to bind to thin filament, and the muscle shortens. And once that's gone, the muscle relaxes itself, and that itself requires ATP as well. And just to show you guys what a skeletal muscle looks under the microscope, we can see right over here, we have these striations, which are our sarcomeres, and we have all these nuclei that are found within the muscle fibers. So that's pretty much it for our video today. Let me know if you guys have any questions or you think I missed anything. And like always, I'll see you guys in the next video. Stay tuned. Have a good one.